So we can officially start. Um, it's my great pleasure to introduce Paul Falkowski. Uh, Paul, we have people coming in from all over the place. Mimi Katz has joined us to hear this talk. Paul, of course, is the Bennett Smith Professor of Earth and Planetary and Other Sciences. He is winner of the Tyler Prize, and he's going to be talking to us about an exciting project today. And I'll let Paul talk about Enigma.
Thank you very much. So Enigma is a NASA program which we developed with Nathan Yee and Vikas Nanda in the medical school, and Bob Hazen and Jana Bromberg and so on. And it contains, if you look down there, there's a little website, enigma.rutgers.edu. You can find all the people that are involved. We have about 40 people that are involved in the Enigma program in one way or another, is either participants and faculty or as postdocs or graduate students and even undergraduate students. So I'm going to begin. The core question is, how did proteins evolve to become the predominant catalysts of life on Earth? And so we have three focused but integrated research themes. And we broke them out into the following way. Theme one is what we call the synthesis of the nanomachines and the origin of life. So that is a protein or peptide synthesis group. So those are the guys like Vic Nanda and Doug Puck and others that design proteins in computers and synthesize them primarily in my lab. Theme two is a bioinformatics theme. It's headed by Jana Bromberg, and it's trying to understand emergent properties, in effect, increasing complexity of nanomachines as they evolved. How did things get to be more and more and more complicated? So we think the earliest proteins were extremely small, and then, of course, they became more and more complicated. We do not have a fossil record of these proteins. So we're having to reconstruct them from a very novel way, and I'll show you that in a bit. And then theme three is headed at Rutgers by Nathan and co-headed by Shauna Morrison and Bob Hazen down at the Carnegie Institute in Washington. And it's the co-evolution of the nanomachines and the geosphere. So we focus on a set of proteins that are called the oxidoreductases. So let me explain very quickly. The Enzyme Commission is a group of people that determines where and what to assign proteins in, and there are 7 Enzyme Commission numbers. So the first Enzyme Commission number is Enzyme Commission 1. It's a very large group of proteins, and they're called the oxidoreductases. So the oxidoreductases are oxidizing and reducing products from substrate. So every oxidation reaction has to be accompanied by a reduction reaction. These are paired reactions. And why do we focus on the oxidoreductases? Because the evolution of metabolism is what these guys do. You have in your body a protein, for example, in your mitochondria, which is the little organelle in every one of your cells except your red blood cells, that exchanges oxygen with the environment. And that protein, for example, contains copper. And it's an oxidoreductase. So it's cytochrome oxidase. So some of these enzymes are extremely old, and they're highly conserved in extant microbes and microbial bodies, such as the mitochondria I just explained. And the reverse engineering is what we're trying to do to understand their evolution, to how these organisms obtained energy on early Earth. Now, let me just explain what gives us some secret sauce here is that Rutgers is the home in the chemistry department of the Protein Data Bank, which is headed now by Stephen Burley. The Protein Data Bank is the largest repository in the world for protein structures by far. There are tens of thousands, there are about 150,000 protein structures that are in the Protein Data Bank. And any one of you that's interested can just go to PDB and download a program which is freeware called PyMol, and you can access any protein you want, and you can see how they look. So the three themes are in this little hokey diagram, which Vic Nanda put together some time ago. So theme 1 is the origin of the nanomachines. Theme 2 is the evolution of them, how they became more complicated. And theme 3 is trying to put this into context of geologic time. So if we consider that the Earth is about 4.56 billion years old, we come to about 4 billion years, and this is the base, of course, of the Archean. There's no sign of life that we can see in the fossil record yet. I think the first initial, and it's very controversial, sign of life is probably around 3.6 billion. Most embarrassingly, almost all of the molecular fossils based on preserved lipids are contaminated by drill oil. And it has been very, very difficult to understand what the origin of the fossils are unless you can have a physical fossil. And this has been a very controversial area. So we finally realize, of course, by the time we get to about 2.5 or so billion, that there is ample evidence of life. And by 2.35 billion, we have, of course, what we call euphemistically the Great Oxidation Event. Now, the way we look at it is a very simple way. We know where we got to. We see complex life around us now. We see microbes all over the place, but we don't know how it got there. So we don't know what was the beginning of the bridge. And that reconstruction is really what the Enigma program is about. It's not prebiotic chemistry. We're trying to understand the gap between how we got rocks to become alive, in effect. Now, I'm just going to go to this very, very trivial slide, the Goldschmidt's plot. And I just want to point out that there are 6 elements that comprise all of the major polymers of life. So proteins are polymers. They contain hydrogen, carbon, oxygen, nitrogen, and sometimes sulfur, but not phosphorus. And the phosphorus molecules are primarily bound up in nucleic acids, which are the machinery that recreates the proteins. And this has been one of the big conundrums in the origin of life is how do you get metabolism that can't replicate itself? And how do you get replicons or things that replicate that don't have metabolism? And this is not a very well scoped out problem to this day. It's been a problem for the last, I would guess, more than 50 years since the discovery of RNA replication. So I just told you that those are what's called the big 6 elements, C, H, N, O, P, and S. And of course, you can arrange them so that they spell the word schnapps. And that's how our students remember that. And then there are at least 54 other, quote, trace elements that are involved in metabolism in life. So I just want to make a very clear statement that this is not a metaphor. Life is literally electric. Every cell in your body has an electrical field across it. You're a walking electric field yourself. In effect, you're a hydrogen storage mechanism. You're deriving energy by oxidizing reduced organic material in the presence, in our case, in the presence of molecular oxygen. So all organisms are deriving energy by moving electrons from a substrate to a product. And therefore, all substrates and products must ultimately be cycled. And so all biological processes are paired. So they're basically half cells. So you can view yourself as an oxidant, an oxidizing organism. And where did you get your the reduced material? You got it by eating food, which is derived from solar energy. So on the surface of this planet, by far, more than 99.99% of the electrons that are stored in reduced products is coming from photosynthesis. Now, we know oxygenic photosynthesis, but long before there was oxygenic photosynthesis, there were anaerobic photosynthetic processes that could produce organic matter. This isn't the only way you can generate organic matter, but it is by far the most important one. Now, in the Archean, so that's from about 4 billion to about 2.5 billion. The primary source of electrons were hydrogen gas itself, which is not in the atmosphere today on Earth, iron 2, hydrogen sulfide, which is still coming out of deep sea hydrothermal vents, for example, and carbohydrates or organic material. Now, it may be funny to think about where did we get organic material? So one of the major sources of organic material in the early Earth were meteorites. And meteorites contain, for example, up to about 100, at least that we know of, 100 amino acids. And they're formed in rock-water interactions in space, no biology necessary. And when they're delivered to the Earth, the meteorites will partially dissolve the amino acids in seawater, and those amino acids are available for metabolism. So those 4 major sources of electrons were the Archean. And until we learned, some organisms learned how to split water, which is the source of the oxygen on the planet today, the major source of liquid water was virtually inaccessible as a source of electrons. So you realize, of course, that, or I'm not saying of course, water has that H2. So when water is split, we never actually make hydrogen gas. You separate the protons and the electrons for reasons I'm not going to go into, but those are recombined later into H molecules that are tacked onto hydrogen carriers in your cells. And of course, the waste product of that is oxygen. The plant is just releasing the oxygen to the atmosphere. Now, I just want to point out, it looks like there's an awful lot of water on the surface of the planet. So we have this, concept that we're 71% of the surface is liquid water. And that's true. But the oceans on average are only 4,000 meters, 4 kilometers in depth. And if you were to take all that water and put it into a ball, that's the volume of the water that would be on the surface of the planet. It's really small. It's amazing that we've had liquid water on the surface of this planet for at least 4 billion years, if not more. And I'm just going to make another little statement here so you can think about this if you're teaching undergraduates. If the diameter of the Earth was to be 1 meter, just take a meter stick, how thick would the oceans be? And it would be represented by 2 sheets of typing paper. That's how thick it is. It's incredibly thin film of liquid water on the surface of the planet. Now, there almost certainly was liquid water on the surface of Mars. It's not there anymore. And we're pretty sure that there was probably liquid water on Venus, although we don't know that with as much certainty, obviously, as we do for Mars. But the three planets, one of them is the only one in our solar system that contains liquid water on its surface today. And it's really amazing because it's one of those paradoxes where the solar luminosity was about 30 to 35% less in the Archean, the beginning of the Archean. The rule of thumb is it decreased by about 1% per 100 million years. And in that case, we still had liquid water. So the disk size, why is the faint young sun fainter? The disk size of the sun was smaller. That's a simple explanation for it. So the greenhouse gas content of the atmosphere had to have been much, much higher in the Archean to allow the liquid water to remain liquid. And of course, at some point, we got to about 2.5 billion years ago, the first global runaway ice house. So we then had solid phase water for most of the planet, we think. Now, I just want to talk a little bit about these rocks. This is Greenland. These rocks are 3.8 billion years old. Greenland's rocks are extraordinarily iron rich, and you can see that these were sedimented from an ancient ocean of 3.8. And you're looking at, obviously, the rusty material is oxidized iron. And even if you drill deep into some of these rocks, you'll find oxidized iron. It's not just a surface exposure issue. It's an interesting phenomenon, which I'll go into in a second. The phenomenon, though, of iron in the ocean led to some thoughts about what would be the reactions of iron in the ocean in the early world to generate an electrical field, for example. And if you take a look at the left side of this slide, you see today at the surface, most of the ultraviolet light is being quenched from the top of the atmosphere. That's the difference between the black line and the red line. And you see also a very funny phenomenon, which most people don't quite understand intuitively. And that is that UV light can penetrate into seawater pretty well. So it's the red and the longer wavelengths lights that are removed very efficiently by water. But the water itself does not really remove, unless there's a lot of organics in it, a huge amount of the UV light. And I just point out why these lines on these why does it look fuzzy? This is not noise. These are Fraunhofer lines. So these are the absorption bands or absorption lines from the corona of our star. It is, for example, taking with iron, removing some of the visible light. And that's how we discovered, for example, the distribution of elements in stars. All right. Now, I'm going to talk about something that's a little old, but we'll get to the new. So several years ago, we had a graduate student in chemistry named John Kim. And in Nathan's lab, he synthesized the mineral iron carbonate. So iron carbonate is called siderite. It was well abundant here on Earth in the early period of Earth's history before oxygen. In the presence of oxygen, iron carbonate siderite is unstable. It it just oxidizes. But here you see the siderite in panel A is the gray-white material. And John burned a little hole in the, this is a quartz cuvette with a UV light, and you can see it's photo-oxidized. You have iron that is now iron 3. And the reaction is pretty efficient, actually. So John spent a lot of time working on this reaction and showed that you can convert siderite, which is in panel A on the left, to a magnetic iron material. And I'm not going to go through this. This is published a few years ago in PNAS. But this is just with light under anaerobic conditions, no oxygen. There's no oxygen here. There's no biology here. This is just light burning through iron 2 to make iron 3. Now, what happens when you do that? The reaction is very complicated. I'm not going to go through the chemical reaction, which we worked out with a guy named Fraser Armstrong at Oxford University. But the point is that you generate hydrogen gas. So you generate hydrogen gas. This reaction only occurs in aqueous phase. So if there's siderite on the moon, it's not going to be photooxidized. But siderite on Mars would create the regolith that's red on the surface of the planet today. Now, what happens when you do this is the carbonate becomes carbon dioxide, and that would go off into the atmosphere. And it's interesting that the surface of Mars has very, very, very small amounts of carbonate in it relative to what we would predict. So I talked to Grotzinger about this when this paper was published in 2013. and it is probably the main mechanism by which Mars became oxidized when it had water. Now, I'm going to talk about a paper that was just accepted last night in PNAS. This is a paper from Winnie Liu, who's a graduate student in my lab working with me and Nathan Yee. And she's done a very similar reaction, taking, in this case, a manganese carbonate, which is rhodochrosite, and you can photo-oxidize it under anaerobic conditions. So there's no biology and there's no oxygen here. And you can make manganite, which is iron 4 in a oxidized form, which is thought to use, thought to be a marker of biological oxygen photosynthesis, oxygenic photosynthesis. It is not a clear biomarker. So manganite can occur because of the production of oxygen. But it also can occur without oxygen simply by photooxidation of rhodochrosite. It was very interesting to try to get this paper published. It was a lot of pushback from people who really want to use manganite as a marker for the formation of oxygenic photosynthesis. All right, now I'm going to skip a little bit. I'm going to want to just make one point, because this is the point which we use to understand if there is life on another planet that is not in our solar system. So the idea from Vladimir Vernadsky, who was a Russian-Ukrainian, one of the very first biogeochemists, actually, if not the first. He wrote a book in which you read in, if you took a look very quickly at the paper that's been sent around. He wrote a book in 1926. It was published in Russian. And it wasn't translated until the later part of the 20th century. Now, certain people obviously can read Russian. Vernadsky's own son was a professor of sociology at Yale University, and he helped translate his father's works. His father never left the Soviet Union, even though he had an opportunity to prior to World War II. But during the Second World War, he was very much involved in trying to develop an atomic bomb. And later, after the war, he was the architect of the atomic bomb for the Soviets. Now, what he wrote in his book, The Biosphere, all living organisms exchange a gas with their environment, which is critical. So if you're going to look for a planetary signature of life outside of our solar system, you're looking for gases that are not in thermodynamic equilibrium with the environment. So they're far from thermodynamic equilibrium. Now, many people realize this, of course, for a long time. Carl Sagan was one of the proponents of this. So many of the core metabolic processes that we look at in the origins of life are related to gas exchange. So I just showed you one possibility without life, and that's to make hydrogen gas. But of course, biology can make hydrogen gas too. Biology can make methane. Biology can make nitrous oxide. That's laughing gas, N2O. It can make nitric oxide, NO. So there are many gases other than CO2, for example, and oxygen that can be potentially used as biomarkers. So I'm just going to very quickly go through an oxidation reduction reaction with you. So oxygenic photosynthesis is trivial. You learned this in the 5th grade, maybe. That your water is being split to make reduced carbon, which is CH2O in biochemistry world. That's just sugar, candy. And the aerobic respiration is you eat the candy and you combine it back with oxygen to make the water and the CO2. So there are two gases that are coming out of your body from your lungs, and that's water and carbon dioxide. Now, obviously, they're not balanced on a planetary scale over geologic time. Otherwise, we wouldn't have oxygen in the atmosphere. So I'm not going to go into this because this is also relatively trivial for the geologists. But you take the candy that is made in the oxygenic photosynthesis and you bury it in the lithosphere. And so it can't be eaten by respiration, okay, by microbes. And that is how you generate oxygen. So without tectonic processes, we wouldn't have oxygen on Earth. It's as simple as that. It's a confluence of not only the metabolism, but the geological processes that allow the accumulation of oxygen by the burial of organic matter over long, long periods of time. And it's not buried simply in the oceans. Originally, it can be around continental margins and shallow seas, but it's because of the Wilson cycle that it's uplifted onto cratons or sent down deeper beneath the diamond belts. And it's preserved in the deep carbon of the Earth and in continental rocks that are inaccessible to aerobic respiration. So I'm going to talk now about the evolution of the nanomachines, what we're really focusing on in Enigma. And I'm not going to go through this, but this is one of Ben Yellen's papers. This is just the pathways, and you can see there are several cycles that are very, very old, which overlap in the centers of things. So what's called the tricarboxylic acid cycle is an ancient cycle that operates in your mitochondria today, but originally it operated in reverse, and it was a photosynthetic process that did not produce oxygen. And then we see these other metabolisms, which are intermediate metabolism, iron, sulfur, proteins, which I'll talk about for a minute, and so on and so forth. So these are the primary metabolic pathways of the planet. All of these evolved in microbes. Animals, as we commonly call metazoans, animals like you and I, or an elephant, or a snake, or a bird, we didn't invent any new metabolism. Nothing new. Plants didn't invent anything new. They're just using the same metabolism as the cyanobacteria, which is the original organism that produced oxygen. And we can arrange these in a group like this. This is a wiring diagram of the major metabolisms on the planet. And I asked Ben to annotate it, which proteins do what. And he discovered that there are only about 400 proteins on the planet that make the world go round, which is amazing. That's a very small number. So we know the sequences from genes of literally millions of genes, but only 400 proteins really are important to make the entire world go round. Now, I'm just going to talk very quickly about this. So the machinery that is splitting water is very, very, very complicated. We don't know it very well. So it's on the left, you see this big machine there, double machine. It's a duplication, a gene duplication. And there's where water is split. And the electrons are passed through the cytochrome, what's called cytochrome B6F complex, which is what is in your mitochondria today. It's cytochrome BC1. And then they're passed on to a second photosynthetic process, which ultimately is generating the reductant for fixing carbon dioxide. This entire machinery contains about 140 genes, or it's encoded by about 140 genes. This is the most complicated energy transduction system on Earth. Very, very complicated. And we know that's the structure of the, for example, the protein that complex that splits water at very high resolution. Very high. I mean, this is a 1.9 angstroms resolution. Many people have been working on this for years. The most active groups are in Japan, in Genren-shen, and Okayama University, and Petra Fromme at University, Arizona State University, and there's a group in Germany that's been working on this as well. Now, if you take a look to the right, you see those manganese-1, those purple balls? The actual machine that is splitting water is composed of a 4 manganese cluster with 1 calcium atom joined by oxo bridges, oxygen. So this is a mineral that is made by biology as far as we know. There's no known mineral like that in naturally occurring. So all of the oxygen splitting processes are accomplished by 4 manganese atoms, which is rather remarkable. So we can array the metabolism of the planet in terms of time, going from more reduced to a more oxidized metabolism. And there's a change in the mid-volt potential of these on the bottom. You'll see that. So the mid-volt potential is a mechanism or a process, a phenomenon, by which we can assess oxidoreductases and what they can do and what they can't do. So if you want to split water, relative to pH 0, you need about a 1200 millivolt potential difference. That's the largest potential difference, in any oxidoreductase on Earth. Relative to pH 7, it'd be about 840 millivolts. So we can array these now in a very simple way. You can look at the entire planetary surface as one big circuit board. So you know the power supply is the sun. You know that there are a series of wires that have to connect the metabolisms of an organism, for example, that's living in the middle of the ocean to a metabolism of an organism that's living in the Rocky Mountains. And what are the wires? Well, we have two major wires on Earth, the atmosphere and the ocean. So the atmosphere is moving electrons around. As you breathe, you're breathing out carbon dioxide, you're breathing in oxygen. The oxygen is not made in the room you're living in. It may not be made in the winter anywhere basically in North America. So you're breathing oxygen that is circulating across the planet that's made on a planetary scale. The oceans are similarly doing the same thing, but obviously the viscosity of the ocean changes the rate at which those electrons can be moved. And then what are organisms doing? So the organisms are basically diodes or transistors, if you will, on the surface of the circuit board. So they're the ones that are changing the redox state of the minerals or the gases and conveying them back into the environment. So that's the way you can think of the surface of Earth. And that's the way we do in Enigma. So then we'll take a look. Can we find the structures of the processes that were the very, very beginning of life? And this goes back to a very old paper by Margaret Dayhoff and Richard Eck. And it was in Science in 1966. We had very, very, very few structures in 1966. And they wrote a very profound sentence that said, the processes of natural selection severely inhibit any change in a well-adapted system on which several other essential components depend. So what does that mean? It means that very complicated machine that I showed you that splits water is the same in a cyanobacteria and in an oak tree, because it depends on a well-adapted group of proteins. You can't just change one protein and everything else remains the same. Now, we took a long time ago, we took a look at what were the major trace elements that were in the oxidoreductases. So if you're moving electrons around, invariably you need a transition metal. And by far, the largest transition metal in oxidoreductases is iron. And it can be in what's called iron-sulfur clusters or free iron, or in the case of, for example, cytochromes or your blood, heme, which just means red. And then the next most abundant is copper, and then manganese and nickel, a little bit of molybdenum, a little bit of vanadium, and cobalt. So this is great if you're in the Archean Ocean. There's tons and tons of iron. But when you get the great oxidation event going and you start moving this further and further into an oxidized system at the late Paleozoic, you rust out virtually the entire iron in the oceans. So the iron in the oceans is now delivered primarily through an aeolian source. That means from wind blowing iron from the Gobi Desert, for example, or from the Sahel in Africa across the ocean, and it's fertilized then because we have continents. If we just had a world of ocean that had become oxidized, iron limitation would have been a major, major problem for metabolism. Now, here is a very, very simple molecule. This is the one that Eck and Dayhoff worked on. It's a 4-iron, 4-sulfur cluster in the center of a small protein. The protein is about 56 amino acids. And I'm not going to go through the spaghetti diagram of showing you what the structures mean. But the point is, we can break this protein was originally a monomer of 28 amino acids. And we focused on, can we make an artificial, very, very, very small type of ferredoxin? What does ferredoxin do? Ferredoxin just ferries electrons. That's all it does. It just ferries electrons across your metabolism. And it's found in everything, everything. Then we can build up on that. We can take and make a much more complicated molecule, which is contains iron sulfur clusters and a little sometimes bit of molybdenum, one atom of molybdenum or one atom of vanadium. And this molecule is responsible for taking the nitrogen in the atmosphere and converting it into ammonia. So it's a reducing reaction. It takes an N2 gas and it makes adds 8 electrons. Ultimately, 6 of them go to the nitrogen, and 2 of them go to hydrogen to make hydrogenase. It's a hydrogenase as well. Now, this molecule is the only one on the planet. Every nitrogen-fixing organism has that molecule, period. It's extraordinarily conserved in structure. And amazingly, it evolved prior to the evolution of oxygen. It's extremely sensitive to oxygen. So every organism that fixes nitrogen must protect the nitrogenase from molecular oxygen in some way. And there are various strategies that organisms use. And then we have other proteins like this one. This is a superoxide dismutase that is based on copper in this case. And this enzyme is responsible for removing the reactive oxygen species in your body. There are many of them. Here's a manganese-based superoxide dismutase. So these molecules are around to essentially protect cells from being damaged by the very toxic peroxides and other reactive oxygen species that are accompanying the oxygenic world. Now, this is an old paper from John Kim. I'll go into a little bit more detail later, but he arrayed proteins according to whether they were sheets or helices or loops. It doesn't matter. Those are the 3 major structures. And then you can draw essentially a vector from point to point to point to point and try to determine what the evolutionary history of it is. And this is basically the evolutionary history of the protein. So at the very left, on the upper left, you see the iron hydrogenase is very, very old protein, and there's ferredoxin. And then in the bottom right, you see the cytochrome C oxidase. That evolved much later. So this is a simple evolutionary tree based only on structure. Only on structure. Doesn't matter where the molecule came from. We're not interested in the bugs. This is the change in the complexity of proteins over long periods of geologic time. Now, Vic Nanda has worked on this problem as well from a totally interesting and different way. We started to break these molecules from structures down into what we would call Legos. So you can see, for example, these iron sulfur clusters forming wires in the left-hand structure. And then you can see in the right-hand structure that these wires are connected to other molecules that contain other metals. And so you break these up into modules where you can say, okay, here's a molybdenum. There are 4 iron, 4 sulfur clusters. There are 3 of them, or 4 of them, or 5 of them. Then a couple of hemes. And we can do this with thousands of proteins. And you come up with many modules. And you can put these into what is basically a map. And these are the Lego sets. The major Lego sets are ones that contain, for example, copper, which is the upper green. That's plastocyanin type molecule. There's one that contains iron, which is in the beta sheets. Those are down the lower left, called symmetrins. And then there's one that is on the lower right, which is the old ferredoxin. That's the one that has the 4 iron, 4 sulfur in those very, very simple molecules. And then the cytochromes. So there's an old rule in bioinorganic chemistry And it's basically, if you are less than 14 angstroms apart, you can transfer an electron. If you're more than 14 angstroms apart, very, very, very little electron transfers will occur. So if you want to make a wire, you have to have these within 14 angstroms of each other, which is a very small distance when you're looking at these very large proteins. And then a former postdoc, Hagai Ranan, who was working with Vic and me, made these wiring diagrams, and ultimately, I'm not going to go through all of this, but found that there are only two motifs, two structures that were at the beginning of life, and possibly they emerged from one. So that's that ferredoxin structure and another structure called a Rossmann-like fold. And I'm not going to go into this because for the purposes of this talk, all I want you to see is we've decomposed using this kind of Lego system, breaking these proteins apart structurally in a computer, not physically. This is in a computer, to find the evolution of these Rossmann-like folds. So, for example, the nitrogenase that I showed you, that's that relatively large, many iron sulfur cluster proteins, has many Rossmann-like folds. The ferredoxin fold is another very, very, very ancient fold that I told you about, and it's found in many, many, many proteins. So there only appear to be two at the origin of life, two motifs, a ferredoxin and a Rossmann fold. So I'll finish off the talk. This is an old paper, again, by John Kim. This is part of his PhD thesis. No, his postdoc. Sorry, he did his postdoc with us. So Doug Pike and others in Vic's lab engineered a 12 amino acid protein that contains what's L and D amino acids, and that's the bottom left. And they inserted a 4-iron 4-sulfur cluster in a glove box under anaerobic conditions. This is very sensitive to oxygen. Now, I'm not going to go through all the nitty gritty here, but this molecule can go hundreds and hundreds of times and transfer electrons. Extremely robust. Extremely robust. So you can make a very, very, very simple amino acid structure that can transfer electrons, which is proof of concept that, for example, very small peptides or proteins could have been at the origin of life, or probably almost certainly were at the origin of life. And later on, other people in the group, John Mutter, who is, I'm John Mutter, Andrew Mutter, John Mutter's son from Lamont, if you remember John Mutter. So Andrew made another simple ferredoxin, and a little more complicated than John's, but it also transfers electrons. And what we're trying to do is make these Legos into hydrogenases today. So we're trying to make a 10 amino acid hydrogenase, which seems to be successful. It's based on nickel. And of course, then if you have these proteins and you have these Legos, you can develop essentially electronics. So you could have an NPN transistor or PNP transistor. You can make a diode. You can make a capacitor. I mean, there's several electronic type of analogs that you could do totally biologically. Now, I'm going to finish off the talk now. I've talked too long, but I just want us to understand that we are launching several satellites, and unfortunately, the limb measurements are very difficult to make. So a limb measurement is when the planet goes in front of its star. We can use the background star as a light source to scan through the atmosphere of the distant planet and try to develop a spectra of the planetary atmosphere. This requires a lot of telescope time, but as we get bigger and bigger telescopes, and we can do many things, we can use Doppler shifts and put these telescopes on land from which you can get much bigger telescopes than you can put into space, even with James Webb, for example. We're starting to try to discern gas phases on planets that are within a few light years of our planet. Obviously, the further out you go, the more difficult it is to acquire enough photons. But over the next 10 or 15 years, I am sure that we're going to find planets that have small amounts of oxygen that are not in equilibrium with the gas phases of the rest of the planet. And we will be fighting with each other in terms of the astronomers, the planetary scientists, as whether that's truly a signal of life. And it's a very, very contentious area right now. But that is where we're at. Is we're trying to derive planetary signals of, for example, oxygen. So this is what the Harvard group is doing right now. Several other terrestrial-based and space-based planetary observers are trying to understand if we have a planet that was in the habitable zone, that means it's close enough to its star but far enough away so it potentially has liquid water on its surface, and it's a rocky planet, which we can infer from other mechanisms, which I'm not going to go into. What is the planetary atmosphere like? And so there are many, many, many thousands of targets out there, and fighting for the telescope time is probably the most difficult aspect of it, technically at the moment. So I'm just going to wrap this up. This is what I would say where we're at now. The first 2.5 billion years of Earth's history, nature invested heavily in the what I call a research and development phase, in which a group of metabolic machines evolved. Now that's the beginning of the bridge. We still don't know how to do that. We still don't know how to do that. All of these metabolic processes evolved in prokaryotes. Those are organisms that don't have a nuclear membrane. So they're in your gut, they're in your mouth, they're on your skin, they're in soil, they're everywhere. There are only about 400 core metabolic machines that make the biological electrons flow across the entire planet world of us, Earth's world, okay? You didn't need 400 originally. Could have done this with a smaller set. The metabolic sequences are coupled on local, but also planetary scales. So they facilitate what I call an electron market between carbon, nitrogen, oxygen, and sulfur, and obviously hydrogen, on a planetary scale. So that circuit board that I pointed to, it's also not a metaphor exactly. It is a global market of electrons based on metabolism. And the contemporary electronic potential on Earth is totally driven by light. We can argue about deep sea hydrothermal vents, but even in the presence of deep sea hydrothermal vents, the reason you have metabolism in deep sea hydrothermal vents today is because there's an oxygen-based water body next to the hydrothermal vent. So they're using the redox potential between the background water of the ocean, which is oxidized, relative to the reduced hydrogen sulfide that is coming out of the vents. So it's still based on the kindness of strangers 2,500, 3,000 meters above you, driven by light. So I thank you for listening, and I'll be happy to answer or share any questions, thoughts. Thank you.